Okay, so hello again, and today we are going to, so I will be sharing my screen, so we'll be going through some slides together, but thank you for being here, and today's workshop is an introduction to mathematical modeling of cell signaling using MATLAB. So my name is Leo Lagunas. I am a postdoc in Dr. Eric Dean's lab. And so for day one, I don't get a lot of chances to introduce myself, but I will talk a little bit about myself. I went to community college where I studied I tried a bunch of different majors, but I ended up studying math. And then I transferred to Cal State Fullerton. So I'm born and raised in Orange County. And I went to Cal State Fullerton where I finished my degree in applied math. And there I did a little bit of undergraduate research in more of a bioinformatics type of lab. And from there, I moved to UC Irvine. And while I was at UC Irvine, I was, as a graduate student, I actually ended up in a signaling lab where we did a little bit of modeling, like a lot of the things that we're going to learn in this workshop. And I did some experiments. So I came from math and I moved essentially into biology/biochemistry. And now here I am at UCLA where I continue to study cell signaling, but now I work a little bit more on protein complex assembly. So I'm sure you maybe have heard other of my lab mates speak, but their projects are more like bioinformatics. I'm more on the deterministic modeling, similar to what we're going to learn in today's workshop. Okay, so why do we even need mathematical modeling in cell signaling? Well, I hopefully don't have to, you are already interested, but to sell it to you in a different way, we've learned a lot about what is happening in a cell from molecular biology, even from biochemistry, and there are two things that are clear. One, pathways get complicated very quickly. For example, the angiogenic signaling network, you can see all of these connections between different proteins that are kind of complicated to unpack. And two, life is not just single pathways, but a network of different pathways, like the regulation of transcription factors in E. coli, where here you can see that some of these transcription factors are regulated by different pathways depending on the pathway that you take. So we can use math to understand the dynamics of these complex networks. And in today's workshop, where we will be learning different building blocks that make up all of the reactions and complicated diagrams. So this is a 3-day workshop. Today, day one, we'll learn building blocks and chemical reaction networks. Tomorrow, we'll learn a little bit about network motifs. And on day 3, we'll learn about signaling pathways and how we can model them. And you will have plenty of practice in each of these days. So here's how the layout of the workshop is going to go. For the first 2 hours, we'll go through, material. Like, for example, today we're going to go through the building blocks, chemical reactions, chemical reaction networks, and then for the last hour, you'll have free time to work on problem sets. So the problem sets are already on the Google Drive, but again, I can share links on here as well. And in that hour, you will have, I'll be here and you can stay on until noon, and I'll be here to answer any questions to help you solve through the problems, or if you want solutions to anything that we do throughout the first 2 hours of the workshop. So let's go ahead and get started. Today, what I hope we will learn or we'll be able to do is to understand the terminology for chemical reaction networks, such as identifying the reactants and the products and the parameters. And second, develop ordinary differential equations for any given chemical reaction, and then include 0, first, and second order, and we'll go through all of these together. Okay, so the very first thing we need to have are some building blocks, and specifically there are some functions we need to be familiar with because we'll see them pop up later, as well as some reactions and their equations, like a chemical reaction and its associated differential equation, and how we can write those differential equations for 0 order, first order, and second order chemical reactions. Okay, so let's just talk a little bit about functions. So there are 3 different functions that I want us to just kind of refamiliarize ourselves with. So the first being a linear function, which you may or may not remember as y equals mx plus b, where m is the slope and b represents the y-intercept. The exponential function, which you may or may not remember as y equals a e to the x, where e is the exponential like number e, and a is usually like the initial value or a separate parameter. And finally, the hyperbolic function, which is usually 1 over 1 plus x. The 1 can change, but usually we have essentially the x in the denominator. So since this workshop is to work in MATLAB, let's take some parameter values and plot them in MATLAB. So what we'll be doing today is we'll be switching back and forth between MATLAB and basically like the slides. So you're going to be seeing my desktop a lot. Okay, here we go. So if you have MATLAB open, you can go ahead and open it. I'll give you a couple minutes to do that. And I've already written a couple of scripts just so that I have everything all together, but we can always start a new one. So if you have MATLAB open, you can start a new script with the new script button on the top left. Oops. Okay. I don't want it like that. Okay, so oops. All right, so all we want to do is, what are we doing? So we are going to choose some parameter values and plot each of these in MATLAB. So plot y equals mx plus b. So in MATLAB works top down, so you have to define everything before it can be used. So for example, we need to define the slope. So let's make the slope, I don't know, 3. We need to define the y-intercept. So let's make that, I don't know, 1. And then you need to define x because MATLAB is matrix based. So we need to define x as a vector. So let's just make it a vector from 1 to 10. And now we can define y. So y is m times x plus b. And then finally, we can go ahead and plot. So let's make this a figure, clear the figure, and then we can go ahead and plot. And on the x-axis, we want the values of x, and on the y-axis, we want the values of y. And then you can hit run. Oh, I have to save it. You can save it however you want, but I'll just save it like that. Okay, and here is our plot. So we expect a line, since this is a linear function, we expect to see a line where the values of x are on the x-axis from 1 to 10, and on the y, the values of y. And you can get fancy and make it look different. So we can label our x-axis as the x values, y-axis as the y values. And then when you run, you can see that the labels are now on the plot. Okay, so take a couple of moments to do the same for the exponential function and the hyperbolic function. And you can change the 1 to any other number you want. And so on the exponential, you can use any values for a. E is already defined in MATLAB, and you can define a vector x. Yes, is there a question? Okay, just take a couple of minutes and I'll be here if you have any questions. Just like 2 minutes. Okay, so let me just share what mine look like. So here are my three graphs. Here is the linear function, here is the exponential function, and the hyperbolic function. And this is a decreasing hyperbolic function. Okay, and again, all of these will be on the Google Drive at the end of the workshop today. Okay, okay, here we go. All right, so let's move a little forward into chemical reactions, specifically from a mathematical perspective. So I just want to go through like a couple of definitions or notation. So this is what like a chemical reaction generally looks like, where we have a term on one side, there's always one arrow, and a term on the other end. And usually there will be a parameter, like, something written up here, at least. If it's not written, it is assumed to be 1 if it's not there. Okay. On the left-hand side, or the left-hand side of the arrow, we have the reactant, and on the right-hand side of the arrow, we have the product, and then the number that is, the value that is written up here is usually, will always be called the reaction rate parameter. So reactant to product with the reaction rate parameter on the arrow. Okay, and there was a paper in 2009 that kind of summarized a lot of the terminology for us in a nice way, where they introduced how we define a node. So a node is like an end of an arrow. So here, all of these are the nodes, they're on some end of an arrow. But then, like, even for example, here we have B plus C, together they make a single node because they are on that end of the arrow. Same with the A plus E, the F, those are all the nodes on them by themselves. And if you have multiple sets of reactions, then we have linkage classes. So here the top is one linkage class, like this top up here, and the bottom is the other linkage class. So essentially like closed loops, or just sets of reactions create the linkage classes. And this paper in particular talks about deficiency theory, which we will not be going over in this workshop. That is something that we could learn later in the future if we want a more advanced intro to modeling. But for now, we're only gonna just learn a little bit of the definitions, which is that we can always have nodes in any chemical reactions, and the nodes are basically made up of the reactants slash products. Okay, so let's go through the differential equations that get associated with chemical reactions. So this is like the second building block, but essentially all we need to model just about anything. Okay, so zero order chemical reactions. So a zero order chemical reaction looks something like this, where we still have a reactant, a product, and the reaction rate parameter, but there's not really a reactant, like the reactant is empty. And which is why we call it zero order, like the order of the reactant is zero, or the power of the reactant is zero. And this is usually used when we see something like A is created at a constant rate. And the biological example for that could be a constant production of transcript, a constant production of protein, or anything that that it is created at a constant rate, or it's happening at a constant rate. And here K is the zero order rate coefficient, or the reaction rate parameter, which does have units of concentration over time. But if we want to know what is happening to A, then we can use differential equation to describe the rate of production. So the way that I was taught was we are gaining A. So, the differential equation for A means that we are gaining A with a coefficient of K. So the differential equation ends up looking quite simple. And in fact, we can actually solve for this analytically. So let me switch over really quick so that we can do it together. So I'm just switching to a different. Okay, here we go. All right, so if we want to find the exact solution, again, you don't have to have taken differential equations to understand this. We'll go through it together. So I'm just going to rewrite my differential equation here. So because A is produced at a constant rate K, the differential equation is just the reaction rate parameter K. And if you have already taken differential equations, or if you're familiar with them, then you know that we can separate the differentials. So these each make up a differential. So I'm just going to separate the two differentials. So dA is equal to K dt, essentially multiplying dt to both sides so that it's no longer on one side. Okay, and whenever you see differentials like that, they are begging to be integrated. So we are going to integrate both sides. So on the left-hand side, the integral of 1 with respect to a is just a. And on the right-hand side, wait, I think I said that backwards. The left-hand side and then the right-hand side, the integral of k with respect to t is kt plus c. Okay, so this is an analytical solution, and it looks like one of the functions we just went over, right? This is a linear function, which means that we can get MATLAB to do it too. So let me stop sharing my screen because now I need the desktop. Okay, so we just went over that. So how do we get MATLAB to solve it? Well, we can actually get MATLAB to solve the entire differential equation without us having to every single time get the analytical solution. You're going to get tired of doing all of this math by hand. So instead, we can get MATLAB to do it. So I took a screenshot here of how we can, like a general outline of how we can get MATLAB to solve it. And we can do it together if you want. Or I can leave this up so that you can try on your own. I think maybe I'll let you try on your own for a couple minutes, and then we'll see what the solution looks like together. So just take 2 minutes to do that. I should probably leave up this, but let me just pause the recording for a second. Okay, I can show you what mine looks like. Where is it? Oh, wait. Here we go. Okay, so based on our analytical solution, which we know looks like a linear function, we expect the solution to look like a linear function as well. So here we have time on the x-axis and the concentration for A on the y-axis, and you can see that MATLAB does in fact plot a line. So we can see that the zero order reaction gives a linear solution, or a linear time course. Time course meaning, because we have time on the x-axis. Okay. So let's look at first order chemical reactions. So first order chemical reactions mean that the reactant is now in the first order. We have at least, one, like a power of 1 on the reactant for, on the left-hand side. Like for example, here we have A being destroyed at a constant rate, converted at a constant rate, or even exported or dissociated at a constant rate. Although it is going to be proportional to its concentration. So for example, if we have transcript degradation, protein degradation, protein dissociation, like, if you have a complex A that breaks up into 2 proteins, that would be a first order reaction. And K is still the first order rate coefficient, or the first order rate constant. It doesn't represent the rate, and it does have units of 1 over time. Okay, so again, let's do something similar. If we want to see what is happening to A, then we can use the differential equation to describe that rate of production for A. So the way that I learned it was, here we have A, but we're losing. In this case, we're losing A, which is why we get a negative. And what we're losing is A that is proportional to its concentration, which is why we get a K here. So even in this case here, we are losing A, so it'll be negative. We're losing A with the rate coefficient of K. So, the first orders tend to look like that. And we can still solve analytically for it. So let me just switch devices really quick. I'm still recording good. Okay. So let me just share my screen one more time. Okay, so let's solve this analytically. And again, you don't have to know how to do this. It's just mostly for you to at least know how it's done, and you will have it in the future if you ever do want to do it by hand. But for the workshop itself, we're also learning how to do it in MATLAB. So we're learning how to do it numerically as well as analytically, at least for the first two. Okay, so let me just rewrite my differential equation. So dA dt is equal to negative Ka. So this is still a separable differential equation, and what that means is that we can separate the parameters. Let me rewrite that. I know I have bad writing, so if there's anything that doesn't make sense, you can just tell me and I'll rewrite it. Okay, so we're trying to separate all the a's to one side, all the t's to the other side. And in this case, the a is here, and we need to move it with its differential, which is over here. So let's first move the differentials, like separate the differentials. So we get dA is equal to negative Ka dt. Now we're trying to get the a's all together, so we'll have to divide a, assuming that a is not 0. So negative K dt is equal to 1 over a dA. And once we have everything separate, like there are no more a's or no more t's in the wrong side, we can go ahead and take the integral of both sides. So we're going to integrate both sides. And I totally understand if these are not familiar to you, but you can you can essentially look up their solutions, but I'm just going to give it to you for now. On the left-hand side, the integral of 1 over a is the natural log of the of a. And on the right-hand side, the integral of negative k with respect to t is negative kt plus some constant c. Okay, but we need to know just a, not the natural log of a. We need to know just a. So we'll have to get rid of the natural log by raising both sides to the e. And now we get a is equal to e negative kt plus some constant c. And rules of exponents say that we can split the exponent such that we have e to the c times e to the negative kt. So I'm just going to let this be another parameter. So I'll call it, I don't know, b, e to the negative kt. So here's on our analytical solution, and it looks like one of the functions that we went over just not even 30 minutes ago. This is our exponential function. So we have an idea of what our solution has to look like. What we need is to solve it numerically. Like we want MATLAB to do it, right? So let's get MATLAB to do it. Okay, so just take, I don't know, let's do 3 minutes, maybe 4 minutes to write a similar code in MATLAB to obtain a plot that will look something like this. And you can let, okay, so A0 is supposed to be B. So let B be 100 and K be 5, and you should get a plot that looks something like this. And I can show you the solution once we come back. So let me just pause the recording while we take, let's do 4 minutes. Okay, I can share what my solution looks like. So this is what my MATLAB code looked like all the way to here. So here's our solution. So yes, it is a decreasing function, a decreasing exponential function, which we did expect. We did see our analytical solution to look something similar to this. Okay. Go. Whoops. All right, let's talk last about the second order chemical reactions. So the second order means we have like 2 reactants. For example, here we have A plus B bind to form C, or, are converted together to form C. The example being, like two proteins that bind to form a complex C. And here we still have the reaction rate parameter. In this case, K is the second order rate coefficient. It's not a rate, and it has units of 1 over concentration times time. So if we want to see what is happening to each species, we can use differential equations that still describe the rate of production for each. So for example, the way that I learned it was, if we want the differential equation to see what is happening to A, then in this case we are losing A, so we should have a negative in front. We are losing A, but it's attached to a B with a coefficient of K. So this is how I learned them. Same with B, we are losing B, so we should have a negative in front of it. We are losing B attached to an A with a coefficient of K. And C, we're actually gaining C. So C looks more like the first order. So here we are gaining C from A that's attached to B with a coefficient of K. And in this case, there's no negative on C because we're not losing C, we're just gaining C. Okay, so let's get MATLAB to do it. So this is our, right before we go on break, just take, I don't know, like another 3 minutes just to plot the solution for A, B, and C on the same plot with these values, like where K is 5, the initials being like 0.5, 0.5, like these could represent micromolar or molar, and you want to start with no C. And you can go ahead and try different values of K and initial conditions to see what is happening to the plot, or what is happening to each of the species. And to help you get started, here's just a screenshot of how we would do that in MATLAB. So we have our parameter and our initial conditions as a vector, and then the system of ODEs is like a matrix or a column vector, where we write the differential equation for each of the species. So this would represent for species A, species B, and species C. And then you can create your time vector, and then you can solve the solution using a similar process as we did for the first two. So just take 3 minutes to do that, and then when we come back, we'll go on a break. We'll go on a break, and then we'll finish the second half, and then you'll have plenty of time to work on any of the problems that we've done so far, or any of the challenge problems from the problem set. So let me stop sharing. I'll pause for a second, and then we'll be back in 3 minutes. I'll show you, a possible solution, or you can share mine. If you have any questions, I'll be right here. Okay, so let me just share what my solution looked like. So we can use any of the ODE solvers, but let me go ahead and plot mine. So here we go. So here we can see that A and B are right directly on top of each other, and as we lose A, we start gaining C. Sorry, as we lose A and B, because we're losing those together, then we can see how we start gaining C. And based on that differential equation, you can actually see it looks like an exponential function on both C and A and B. A and B are decreasing exponential, while C is an increasing exponential function. So you can continue to play around with the parameters and see what happens. What if A and B start off at different levels? What happens to C? What if we have no B? What do you think will happen to C? And you can ask them a lot of those questions by just changing one or two of the numbers that we have. Okay, so now that we're back, we're gonna go ahead and take a break, like an actual break, for 10 minutes. So it is now 9:55, so we'll come back at 10:05, and we'll finish the rest of the material, and then you'll have plenty of time to go over the problem sets that we have. So go ahead, take a real break, go do something. I'll go ahead and pause now. Okay, welcome back. I hope you took a break. All right, let's go through the second and last part for today, where we're just going to go over chemical reaction networks. then we'll take a break and then you'll have plenty of time to work on any of the problem sets for today. Okay, so let's talk a little bit about steady state analysis. So we saw before how we can get an exact solution, and we can start asking questions, if the system does have an exact solution, like an analytical solution, can we learn something from it? And steady state analysis helps us kind of understand a little bit more about what is happening, in terms of the species or the items in the chemical reactions, such as like, do we have a steady state? What parameters are controlling the steady state? And of course, can we use algebra to find actual exact equations for these steady state values? And if so, can we learn something from them, such as stability? We're not going to go too much into stability analysis, but just an introduction so that we are aware about what it is. So how do we actually find a steady state? So a steady state is a condition or, a state of a system in which nothing is changing in time. So in other words, when the rate constant is 0, and that happens when the derivative is 0. So here we have this equation for, this was for the first order chemical reaction. We can find a steady state by setting this equal to 0. So let's just go through a quick example. Let me just switch devices so that we can write together. Where's my pen? Okay. Okay, so how do we find the steady state? So we can find the steady state by setting the derivative equal to 0. So we would have 0 equal to negative Ka. And then we have to solve for each of the species, for the parameters, for everything. So in this case, we have a pretty straightforward solution where either K is 0 or A is 0. So our steady states would be whenever A is 0. The parameter equal to 0 is not going to happen simply because we have an equation, where, if we have this reaction taking place, that means K is not 0. Okay. Any questions about setting, solving for steady state? Okay, let's try another one. Let's try and find the steady state for the second order system. This was one where we have A plus B goes to C, like that. That was this one. So we would do the same thing. If we wanted to find the steady state for A, then we set the derivative for A equal to 0. And we would solve this in the same fashion. Either A is 0 or B is 0. So these would be our steady states. And we put an asterisk. I think that is common to put an asterisk to signify steady state. I've also seen a steady state equal to 0, like that, or B. My apologies. There's just a phone call coming in. Okay, and that's how we would solve for steady state. Same with C. If we wanted the steady state for C, we would set the derivative of C equal to 0, which is just K times A times B, which was the positive one, and we would solve in the same way. So A 0 or B 0. So whenever we have no A or B is when we have a steady state. Okay, so let me just switch devices again. Okay, we did that. Okay, so let's talk about a very simple model of gene transcription and mRNA accumulation. So, we can ask a little bit more involved questions now that we have the foundation to how we can write a differential equation if we have a chemical reaction. So for example, here, if we wanted to know how does the amount of an mRNA vary as a function of the transcription rate and the mRNA degradation rate, which means we have both production and degradation. So here we can see that if we, Y representing the transcripts or the gene, the gene that we're looking at, it can be produced in a zero order fashion and degraded in a first order fashion. So, we can, we make Y constantly, and there's a first order degradation or decay. And if we use the specific parameters, then we have, values for both P and R. So here P and R are, replacing the K. So instead of a K, we just have a P, and instead of a K, we just have an R. So here we can write the differential equation that's associated with the production of Y, or how much Y we have over time. So if we write the ODEs, let's start with this left-hand side here, which is a zero order production. Then if you remember the differential equation from before, we are gaining Y with a reaction rate parameter P, but we're also losing Y, so that's why we have a negative. We're losing Y with a coefficient of R. So that's the second term. So you can imagine that if you have a lot of equations, you're just adding them all up, which is why they're ordinary differential equations. Okay, so what does this time course look like? So we can try and plot this in MATLAB. So we can do it together, or you can try it on your own. Would you like to try it on your own for 3 minutes? Let's do 3 minutes. Okay, you'll have more time to work on that specific problem in the last hour of today's workshop. So let me go to the next part. Let me just share my screen really quick. Okay. So if we wanted to find the steady state of this system, this particular system, I do have the solution here written out just so that you can have the slides later. But remember that when we're finding steady state, let me just rewrite my differential equation. What we're doing is setting the derivative equal to 0. So we have 0 is equal to P minus R times Y, and we want to solve for Y because we're looking for the steady state for Y. Or you can do the star as well. So if we want to solve for Y, then we would add RY to both sides. So we get RY is equal to P. And we want to get Y by itself, so we would just go ahead and divide R. So Y steady state is P over R, which means that what we have is a dependence on both the degradation and the production parameters. So it depends on degradation and production parameters. Yep. Okay. Okay, so let me switch devices one more time, just because I like the one on my desktop. Oh wait, am I recording? Yes, okay. Okay, so let's look at the interconversion reaction, which now, we've seen these before where you have arrows going in both directions. At least a lot of times when we see models in papers and in the field, we are actually seeing this to be a common setup where we have an arrow going in both directions. And the interconversion reaction is actually a quite common one where it can represent things like a protein that is isomerizing between an open and a closed conformation, such as like hemoglobin, when it's bound to oxygen, and then it kind of becomes an open conformation. When it's not bound to oxygen, it's in a closed conformation. There's also the ion channels that open and close. We can think of it as a biomolecule that moves between two cellular compartments, such as from the cytoplasm into the nucleus, and therefore from the nucleus into the cytoplasm, back and forth. It can also be used as a simple model of phosphorylation or modifications, protein that is modified versus a protein that is not modified, and it can switch between the two states. And here both the K forward and the K reverse are the first order rate coefficients, and they do have units of 1/10. Okay, so the ODEs, we can write them together. I've written them here for us so that we can use them already. But if we look at both, let's start with A. With A, we are losing A with the coefficient of KF or K forward, but we are also gaining A from B. So we're, which is why we have a plus. So we're gaining A from B with a coefficient of KR. And then if we look at B, we are gaining B from A with the coefficient of K forward, but we're losing B with a coefficient of KR, which is why we have the negative here. And if you look at these closely, they're just negatives of each other. This is the opposite of this one. Here we have a negative KF, while for B we have a positive KF, and same for the B component. So you can see that there's some symmetry happening here. So let's try and do this in MATLAB so that you can plot a time course for both A and B. Oops, sorry. And and what do we see with the time courses? So we can do this together, or we can try it by ourselves. Let's try it and by ourselves for just a couple of minutes, and then we'll come back and I'll show you what mine look like. Let me pause for a second. Okay, let me show you what my solution looked like. So we're looking at the interconversion example, and here we have, KF and KR, I made them to be the same with initial conditions where we only start with A and no B. And, here's our system of ODEs, time vector, solution, and then we can go ahead and plot. So let's evaluate it, and you can see here that we start off with some A, but as we lose A, it'll convert over to B. So now we have B increasing, and you can play around with the parameters. So let's say K reverse is bigger. Let's make K reverse 10. What do you think will happen? So let me make sure that my plot is here. So now you can see that there's a steady state for both, where we don't get a complete conversion over to B. We don't lose all the A over to B. What if we do the opposite? What if K forward is the bigger one? Oh, now you can see that we are losing almost all the A over to B. So almost all of it. And you can imagine that as you play around with those further, you can, watch how A and B are influenced by just those two parameters. Okay, and finally, let's go through a binding reaction. So let's look at the example above, or the reaction here. We have A and B binding to form a complex AB with a forward parameter of K on and a reverse reaction K off. So if we look at these, we should be able to write the ODEs for each of the species and ask ourselves the same thing. What is happening to A, B, and the complex AB over time? So I should probably switch devices just so we can write the ODEs together. So I just want to write the ODEs so that we can write them together, and then you can have these in the future. Okay. So what do these reactions look like? So let's start with A, the differential equation for A. dA dt. I think I need to make the pen thinner. Let's try it. dA dt. So here we are, let's see, we are losing A. So from here we're losing A. So that means we'll have a negative. We're losing A. It's attached to a B with a reaction of K on. So we're attached to the B. Okay. And then the reverse reaction, we are gaining A from the complex AB with the reaction parameter of K off. So this is, I'm going to put this in parentheses because it's the complex AB. It's not A times B. Okay, what about B? dB dt. We expect that to look almost identical, right? Because we are losing B. It's attached to A with the with the parameter of K on, but we're also losing B. Sorry, we're gaining from this reaction, this one. We are gaining B, so it's a plus with a forward parameter, with a parameter of K off from complex AB. So it has to go in parentheses because it's a complex. And finally, DAB, which is the complex. That one, you can see here that we're gaining it from this reaction. But we're losing it with the parameter K off. This is AB. So this would be our set of ODEs. So that, that's this part. Now, what does the time course look like? And if so, can we find a time course? I'm sorry, can we find a steady state? So let me stop sharing really quick. So let's try that in MATLAB really quick. Let's try to solve in MATLAB this set of ODEs. I can pull them back up if we need them, but it might be easier if we look at them here. This is just how I started. And you can go ahead and try that on your own and see if you get a similar result to what I got. And again, all of these solutions will be up on the Google Drive as soon as the workshop is over. So we'll try that, and then we'll take our final break. And then when we come back, so I'll stop the recording here. And for the recording, we won't see after the break, because after the break, you're just going to come back and work on the solutions, the problem sets. So I can actually look at those. Where did I put them? Where did I put them? How do I move that? Give me a second. I put them. Here we go. So here's the folder. You go to problem sets, and then here are the problem sets. So for day one, this is everything that we did together all the way to here. And then these are the ones that you can work on for today all the way to, so it's just 3 problems, 8, 9, and 10. Those are the ones that we'll do this problem, take a break, and then come back. You'll have the remaining time basically until noon to work on these problems, and I'll be here to answer any questions and go through any of the solutions that you might need. And then in here, in the same folder, I'll add in today's MATLAB script so that you can have access to it. So I'll go ahead and stop the recording. Let me stop sharing really quick. So I'm going to pause the recording, so then from here on you won't see the recording until tomorrow.